So this week is about development, so childhood development, in particular looking at nature-nurture, the roles of genes and environment in creating cognition and creating the brain. And obviously the nature versus nurture kind of question is one of those that is kind of central to psychology. A lot of people come to the degree because they're interested in this. And here this is around obviously individual differences, what makes me different from you and so on. We have different genes, different environment, which one counts the most. And of course this is very intuitive, but it's also a little bit too simplistic. And again, people have kind of vacillated historically over which one accounts for most of the variability. So originally Galton was one of Charles Darwin's cousins, and he was interested in applying the theory of natural selection to cognitive traits, and he argued that things like genius or cognitive ability was a product of nature. Obviously the kinds of people he studied would have very different environmental experiences in terms of the quality of school they went in, so there was a lot of confounds. But actually he did do proper scientific research as well. He invented the twin study, for instance. So the nurture debate was kind of very common in the early 20th century. So Freud, for instance, would talk about everything was related to your early childhood experience, your parenting, and so on. And the classic example here is Skinner's kind of behaviorism, the idea that all of our traits are kind of learnt stimulus-response pairings that have been reinforced, and so on. The middle ground with this is really kind of Piaget and his followers. So Piaget is a Swiss psychologist, I think, in the pre- and post-war period, mid-20th century, who basically took more of a middle ground in terms of nature versus nurture. And essentially what he considered the child going through was a series of stages. In effect, what this kind of constitutes is that the creation of a stage is something that is somewhat inbuilt in the child or the infant, so their brain is ready to learn or ready to acquire certain information. But in order to actually proceed or mature onto the next stage, it requires then an environmental input. So without a suitable environment, in effect, children become, to some extent, fossilized at early stages of cognitive development. So in effect, you've got the genes creating a readiness to learn, then you've got the learning, then you move on to the next stage, which is then kind of either more maturation of the brain or further learning episodes and so on. So he described it in terms of this kind of cyclical feedback. So modern approaches are not completely dissimilar for this, but they would not dissect development into these kind of discrete or hard stages in the way of this. They would postulate it as more continuous, but nevertheless the idea of this kind of cyclical idea between readiness to learn and your brain's capacity and what's there in the environment is kind of common there. And here the emphasis is more on kind of brain-based constraints, what your brain can do, and it can do certain things maybe in the first few years of life that it cannot do later, and so on. This is a useful way of kind of rethinking brain development from the kind of very traditional approach, in that one kind of imagines that the genes creates your brain structure then determines how it functions, and your functions determine your experiences, so how you interact with the world, the jobs you get, this sort of thing. That's what I mean by experience here. This is seen as being the kind of the correct or more nuanced way of doing it, in that basically there are arrows in both directions. So obviously you need a certain amount of genes to create the brain structure, to create the brain function in the first place. There can be no experiences without some biological reality. But at the same time, you can have the arrow going all the way back down. So these are things such as, for instance, epigenetics. So whether you had a stressful childhood can switch on or off certain genes and the way that genes operate, for instance. And that's one of the examples I will go through. So it's certainly not the case that the genes kind of create your state of the brain when you're born and then you wave goodbye and say thank you and so on. That they are continually there to kind of grow and as part of everyday experience. So for instance, creating synapses as we learn and so on is partly under genetic control. So I'll talk about the structural development of the brain, the functional development of the brain, sensitive periods, so whether certain things are innate, and then we'll come back to individual differences and looking at modern twists on kind of the nature-nurture problem, how genes and environments actually interact. So prior to birth, there's a rapid increase in the number of cells. So stem cells form neurons that then kind of differentiate into neurons in what's called a neural tube that in effect forms the spinal cord and then bulges at the end into the brain. And these neurons in effect kind of find their way in the brain by various means. So some of the neurons literally just get pushed to the surface as new ones are formed underneath, but they in effect grow, and the axons grow according to in effect the soup of chemicals that are in the brain. So you get molecular gradients that, for instance, tell you where the frontal lobes are going to be. If a molecule is very concentrated, say in the anterior, it determines that these neurons will connect to other regions in a particular way, and ones at the back of the brain will connect, for instance, to circuits in the eye, and so on. So you've got these molecular gradients that kind of determine how and where the neurons are kind of wired and how they kind of migrate through the brain. Either pushed to the surface or carried to the surface on what are called microtubules. Within the last few months of life, you start to see the kind of wrinkled surface of the brain, and when an infant is born, basically it has pretty much all its neurons in place. So what is growing as the head grows is all the other stuff. So this is kind of adding myelin to the axons, which happens all through development until early adulthood. And then the other things in the brain, so the vasculature, the glia cells, the support cells, and so on for the brain are kind of added, and this is how the brain grows. In terms of what the role of the genes are, it's basically that the connectome, if you will. So by this I mean kind of the pattern of synapses. So we have 86 billion neurons with 10,000 synapses, and this is millions of times bigger than the genome. So what this means is the genome cannot specify how the brain is wired at that level of specificity, because there just is not enough information in the genome to do that. So what the genes are doing is not creating a fine blueprint. The term blueprint's misleading. What it's doing is creating guiding constraints as to how things are going to appear in the brain. And in effect, we can think of the kind of structure of the brain being kind of emergent properties of other constraints that are kind of genetically encoded. So, for instance, your genes might specify how many cell divisions your kind of neuroblasts go in order to create how many neurons you have. And what you find is that, for instance, if you have genetic mutations in mice, that they will have, that creates more neurons, they will start to get this kind of wrinkly surface. So what that means is that there is no genes for having a wrinkly surface. There are genes that tell the brain how many neurons to grow, and then more neurons, you've just got to pack them in, and the surface will wrinkle under its own force, in effect. And similarly with regards to other things that determine the shape of the neurons. So the idea here is that you've got this kind of folded sheet, and then under it you've got your axons, your kind of wiring thing. But you could imagine the axons as being like elasticated, and they are literally pulling the surface of the brain into shape. So in this way, what you would have is genes that kind of determine the orientation and the elasticity, if you will, of certain axon bundles, and you get the shape of the brain for free. So there are no brains that code for the shape of the brain. There are brains that code for how certain major axons are laid out, and the axons will pull everything else into shape. What this also means is that identical twins do not have identical brains. They have very similar identical brains, and that's simply because having the same genome won't create exactly the same output. It's like kind of baking two cakes from exactly the same recipe. They will be broadly similar, but they will not be identical. They will have different patterns of bubbles if you look in the microscope, and so on. And that's in effect what the difference between identical twins are. You're just running the same genetic program twice, and you will get minor variations. And it's not just the environment, it's just that the genetic program is not fully specified. What you would also have is that there would be kind of spontaneous firing of neurons. So even when an infant is in the womb and it's had no real patterned light activity, obviously it will get a small amount of light through the womb. In effect, what you have is that the retina generates electrical signals through it, not triggered by light. But these electrical signals would then propagate to the lateral geniculate nucleus and your V1 and so on. So in effect, what's happening is that these pathways are strengthening themselves just by creating electrical signals and passing them on, even in the absence of anything in the environment. So it means that your primary visual cortex has been trained to accept light input just through the spontaneous patterning of neural activity. What this also means is that the brain can be quite flexible in terms of how it grows, at least within certain parameters and critical windows. So this here is somebody who was, so this is a 10-year-old girl who effectively never had let me get it right, a right hemisphere. But the question is, a lot of these studies, when they're scanned, it's quite surprising, that people have literally kind of taken people and discovered that they are missing half a brain, and they can be, in many respects, not necessarily cognitively normal, but certainly not having the level of impairment that the textbook tells you they should have. In this case, this was a study of visual processing. And in effect, what you have here is that normally the right side of the brain would get visual information from the left side of space, for instance, whereas here there is no right visual cortex. So what you find is that the other hemisphere gets information from both sides of space, and that's what's shown here. So the green and blue is the right visual field, but you also get it from the opposite side of space as well. Shown here in yellow and red. And here the tracts, the major white matter tracts, are reorganized here at that particular time. Here she'd had a problem in the womb. I think this was kind of known at the time of birth, but there are these cases where it has not been known until quite late in childhood that they're missing half a brain. What about after development? Well, after development, basically what happens is that your cortex goes through a kind of a maturation process, which actually is to do with reducing the number of synapses in your brain. So the time of your life that you had the most synapses of your brain was the day that you were born, pretty much. And that's what's shown here. So this is looking at the number of synapses in small regions of human brain. Here got post-mortem. So this is an MRI or so on. This is literally taking slices of brain and looking at them under suitable electron microscopes with that. And every part of the brain shows this characteristic rise and fall, which means that the time of your life that you had the most synapses was early on, but whereas some areas kind of mature earlier, so for instance visual and auditory cortex, your visual cortex reaches mature levels within the first few years. Things like your prefrontal cortex don't reach maturity until late adolescence. So again, not dissimilar from your age group here. What's going on here is obviously, you kind of imagine that losing neural connections is a sign of pathology. You kind of imagine that this is like a proto-Alzheimer's, but it's not. What this is kind of an efficiency saving or tuning into the properties of your environment. So there are lots of examples. For instance, the classic one, that infants can discriminate phonemes that aren't in their language. So Chinese babies, for instance, can hear the difference between R and L, whereas a Chinese 3 or 4 year old struggles to hear that difference. And here, that is partly what is going on here, that you're tuning into your environment and that this is healthy, and you're tuning out of aspects of your environment that you don't need. And this is also related to sensitive periods and so on, that it might mean that actually if you learn English as a Chinese person later in life, this is going to be a hard skill to acquire, that you do not kind of get these back as quickly as you would imagine. But you can have the reverse happening as well. So although your brain is kind of reducing the number of synapses, you can increase your grey matter by becoming skilled at certain things as a result of your environment. So this is your environment triggering growth in the brain, and probably at the level of synapses. And this is a study done over 3 or 4 months where they teach people to juggle. And in effect, they look at how much grey matter, like the thickness of the grey matter in various brain regions. Here it's in the part of the brain involved in detecting visual motion, V5, and a part of the brain in the parietal lobes involved in hand-eye coordination. I think it was LIP, but it was certainly one of those kind of parietal regions. Here, this is the original grey matter density, and this is after they've been trained in juggling. And then this is after they've been stopped training. So unless they're carrying on juggling, most of the people do not carry on juggling every day after the experiment. But nevertheless, I think this is several months later, they still are different from the baseline as it goes down. So here they're not necessarily growing more neurons, they are probably proliferating the number of synapses or increasing the support within the grey matter. That might be the blood vessels in the grey matter and so on. And we've seen the example of driving a cab in London, where the idea is that it is the experience causing the brain structure to develop. And what causes the brain structure to develop is switching certain genes on that will regrow your synapses. So it is working its way all the way down to the genetic level when you're doing that. So this can work both ways. So you're losing your synapses, but your experience can regain them as well. What about at certain times in life? Does it matter when you acquire a certain skill? So this is related to the idea of critical or sensitive periods, that basically that you need to be exposed, for instance, to language at certain times of life in order to learn, and then at other times you won't necessarily be able to acquire it. So Lorenz was a researcher who basically studied young birds. So I think that they were some of the chickens or geese and so on, I think were his main participants. And basically what they find is that when a baby bird hatches, it will follow the first moving object it sees. And if the first moving object is a human professor, they will literally just follow him around the yard and so on, as if they think it's the mother. And what was found here is that there was a particular time window in which this was to happen. And basically, if they do not see a moving object in that particular time window, then the behavior kind of drops out. So this suggests what was called a critical time window, that unless you kind of see a moving object within that period, you will never, what's called an imprint, you will never show this kind of parent-like attachment. Other research suggests it's more of a sensitive period. So the distinction between a critical and sensitive period. Critical means that it's like a kind of, that's called a square wave in a thing, that a cliff edge at which point you simply cannot learn. A sensitive period means that there is a period in which learning is enhanced and a period in which learning is hard but not impossible. So it's more of a kind of a soft difference between them. So even with here, you can, there is evidence that you can kind of modify this. So typically, once you've imprinted the first moving object, you won't then start following your real mother, but you can do it if you have a series of stages. So if you've got kind of fake objects that are gradually morph from one into the other, you can develop a training routine that will make them follow B from A around. If you've got A that's a little bit like B and then a little bit more like B. But again, this is a long process to get them to retrain to do that. So it can be done, but it's not straightforward. In vision, the evidence, this is kind of Colin Blakemore's kind of classic and somewhat controversial research in the 70s, that basically cats who have visual deprivation, that basically there is a critical point at which if the animals cannot see light, that they do not develop their cortex in quite the same way. So here what they're looking at is things like orientation selectivity, that neurons respond to lines of particular orientations and they fine-tune them in there. So there are certain periods at which that will happen, or also developing binocular vision as well. So this is being able to integrate input from two eyes, for instance, that there are particular periods in which that can occur. But at other times, you can kind of blindfold or whatever, and it kind of comes through. In humans, obviously, you can't do this experimental research, but it has been done, for instance, in people with cataracts. And it's been shown that, again, when you kind of remove the cataract, that there are kind of sensitive periods, but again, not necessarily critical periods in which this is happening. What about language? So an original claim here is that adolescence is crucial here. But again, the problem with language is that it's not a single skill that you have in your brain. It's made up of multiple skills. So, for instance, detecting phonemes is not necessarily something. It's something that would mature at infancy rather than adolescence. Things like grammar, people have looked at. So there's this rather horrible case of Jeannie, who was effectively locked in a cupboard in Los Angeles until she was a teenager and her parents didn't talk to her. And basically at the time it was found that when people did start talking to her, language increased very rapidly. But then it never reached normal levels, in effect. So initially it was taken as evidence that actually you can acquire sophisticated language and grammar in that period. But because it never reached the ceiling, then people kind of revised that. But here the problem, of course, is that there's an awful lot going on in somebody's brain if they've been locked in a cupboard, all their social world and so on. So it's not just language that's going on here. But what you can look at more experimentally is you can look at people who've acquired a second language, either in childhood or in adulthood, and see how their brain differs. And here there is a suggestion that critical and sensitive periods are important. So obviously you can learn a language in adulthood, but you might not be using your brain in exactly the same way as people who learned it in childhood. Here, in effect, what this is looking at is that it's subtracting away, I think it's grammaticality judgments in two languages. And in one case, you're subtracting away the second language from the first language. So here it looks in people who were bilingual from birth that there's nothing going on in their brain. That's not true. All it is that your first and second language are equally as efficient as each other. So when you compare your French versus your English, they are using exactly the same brain mechanisms, and it looks like there's nothing happening in fMRI. But that is not the case for late bilinguals, that basically their second language nearly always has a different kind of pattern in these kind of frontal areas, including Broca's area here. And it is to do with when it was acquired rather than the level of proficiency. So these people here have high proficiency and these people here have lower proficiency. So it is, when you can separate out proficiency from age of acquisition, it seems to be the age of acquisition that matters more than the level of proficiency in this case. So again, this suggests that there's something of a sensitive period here. But the sensitive period here is observed in terms of the efficiency of the brain networks, because a lot of these people would be very good second language speakers, but their brain is just having to work harder to process, in this case, grammar. There's a study in the journal Cognition by Steven Pinker where he has a sample size of, I think, 1/3 of a million, and he looks at people's kind of grammaticality judgments in other languages, and he also kind of argues that basically there's kind of a peak within kind of adolescence beyond which it becomes very hard to get more efficient grammatical judgments. So that's worth having a look at. You won't see many papers with a sample size like that. So what is the mechanism for this? Basically, what the idea is that there is kind of some kind of readiness for learning in this kind of Piagetian sense of the word. And what this probably is this kind of exuberance of synapses that are there just waiting to see this first moving object, or waiting to kind of acquire rich linguistic stimuli and so on. And then when these stimuli are acquired, what then happens is that there is kind of a reduction in the unused synapses, or a reduction in plasticity of them. So at that point, they cannot reconfigure towards new languages or towards other things. And you would have different windows for different skills in different parts of the brain. Basically, there's evidence that either the closing of this window either occurs genetically, or that the closing of the window is triggered by the environment. So the environment decides actually that these stimuli, that this brain mechanism is now in place, and the environment shuts it off, in effect. And there is some evidence of this, that basically these windows can be somewhat flexible, that they will carry on waiting for a stimulus, but they won't wait forever. So if you don't have your cataract operation at the right time, you will never have a fully functioning kind of visual cortex, for instance. Does it make any sense? So is it the case that you can have any kind of innate knowledge? What does it mean to say that the brain of infants or whatever has something that's innate in it? Here, there are kind of two ways in which people use innateness. One is, for instance, as an instinct, and what they mean by innate here is that the behavior is being selected by evolution. So again, Steven Pinker has a book called The Language Instinct, in which he effectively argues that language is innate, which seems like an odd argument, because obviously you could be born in France or England or China or whatever, and everybody speaks different languages. So here, that is not denying a role of environment. The environment is important, and if quite frankly, if you're locked in a cupboard, you will not develop language because you don't have the right environment. You have nobody to talk to in that case. So here, the term innate means that there is a predisposition towards certain things, but it does not rule out the role of the environment. And this definition of innate is not so controversial. The second definition of innate means that things happen in the brain or cognitively without the need of any environment, and that this is more controversial. So the kinds of claim here is that the brain kind of is, or neurons in the brain are tuned to detect faces even before they've ever seen a face, for instance. That would be an example of innate knowledge without any kind of environmental input. But there is some evidence of kind of innate knowledge, and this is the kind of the orientation tuning in the cat's visual cortex here. So what you can see here is the tuning of individual neurons to different orientations. And basically what you find is that in the first 3 weeks, it doesn't matter whether you've been exposed to light or if you've never seen light. Your visual cortex matures and you get this kind of clustering together of neurons that respond to different patterns of light orientation when they're tested, even though there has been no light in the environment. So developing neurons that are tuned to particular things in the visual environment does not necessarily depend on vision. But then maintaining that does require vision. So without vision, basically things go backwards. So at this point here, between 21 days and 45 days, if you do not get your vision, in effect, you take a step backwards in development, that you will have that. So again, this is the idea of kind of readiness, but you can have neurons that are tuned to particular properties, face-like things or orientations without ever having seen an oriented line in your life. Your neurons are ready to see them, for instance. So this is kind of consistent with some definitions of innate. Obviously it's not, being born to speak French or whatever, but your neurons are there to detect certain kinds of information. It is not a blank slate. There are other examples of innate knowledge in this sense as well. So there's a phenomenon called prepared learning, or readiness to learn, and this is the idea that you become phobic for some stimuli and not others. So you can condition people, for instance, by giving them little electric shocks to become phobic to snakes and other things. And here, obviously, not phobic in the clinical sense, but in the sense that they will, for instance, show fear-like reactions when they see them that you can measure, for instance, in heart rate. And so on. But you don't necessarily get that using the same experimental paradigm to flowers or chairs and other things. So it's as if that in your brain, it's not that you are born to be scared of snakes, but you're born with a disposition or a potential to be scared of snakes. And then you need the environment to set that in place. Okay? But it's not there for all kinds of stimuli. It's hard to become phobic to certain other stimuli. And that's Seligman's idea of prepared learning. So again, here, it's not that fear of snakes is innate, but the disposition to acquire a fear of snakes is innate. So it's a different kind of logic. But again, infants, if they're given bitter tastes, will pull a kind of a disgust face, and a sweet taste, they will do this kind of thing here. So again, that is not something that's learnt. It is something that they are coming to the world with, is a preference for certain stimuli. And again, here, things like attachment in infants. So again, the example of the goslings following the professor is an attachment. In kind of monkeys and humans, people have looked at kids who've been separated from their mothers and put into orphanages, or experimentally, Harlow did this with monkeys, for instance, and shown how there are kind of sensitive and critical periods with this. But the idea behind that is that there is an innate response to seek out parents and to have a limited number of caregivers in effect. So it's not just a learnt response, for instance. So in the behaviourist tradition, loving your parents was a learnt response to having your needs met. Whereas in this framework, infants seek out and want to love their parents, even if they're not very nice parents, in effect. Okay, so for the final bit, I'll turn back to nature and nurture. Here, looking in particular at how genes and environment interact to create differences in cognition, brain, and also mental health. So the traditional approach for disentangling nature and nurture was to use things such as twin studies and adoption studies. In twin studies, in effect, what you're looking at is how similar identical twins are relative to non-identical twins. So identical twins have 100% of their genes in common, and non-identical twins have 50% of their genes in common. So what you find is that the difference between identical and non-identical genes gives you an idea of the degree of heritability. The assumption there is that each twin has roughly the same environment, which of course is roughly true, but not exactly true. In adoption studies, in effect, what you're doing is that you are taking the same environment. You're comparing, for instance, biological children from against adopted children, for instance, and seeing who is more similar to the parents. In some cases, you can maybe there were tests done of the adopted away parents. So before they gave birth, the parents would fill in various questionnaires, and there were particular data sets, particularly around the 60s and 70s, where that was done, where that kind of adoption practice. Basically, mothers used to go away and have their children in private because it was too shameful, and they would just be bored stiff, and the psychologist would just go and give them loads and loads of cognitive tests. So they had good data from the mothers, and then they would wave goodbye to their children. But basically, what you can do by comparing either related and unrelated people within the same family, or twins that differ in terms of their genetic ratedness, is that you can compute something called heritability. So heritability is a measure of the degree of variance in a trait in a population that can assign to a genetic rather than environmental difference. So it is to do with the amount of shared variance that something's explained. So what that means is that if something is heritable at the rate of 0.8, you can't necessarily say it's mainly caused by the genes, because it might be that even if that you still need an environment for it to happen. So it's not the case that genes are deterministic, that it makes a trait inevitable necessarily. So to give you some examples of how not to interpret this, then things like the heritability of reading ability is going to differ depending on your culture. So if you go to a culture where everybody is taught reading to the same level, everyone goes to school, then reading ability will appear to be strongly heritable. Whereas if you go into some cultures, I don't know, maybe in South America or Africa, where some children have good access to school and some have limited access to school, the reading ability will not appear to be heritable. And that obviously says nothing about genes. All it means is that some children don't go to school. So the thing that determines who's good at reading and bad reading is your wealth and not your genes. It's because both of these things have to add up to one. So it's not the case that genes are less important. Well, the basic genes will determine people's reading ability in all the countries of the world, but what determines heritability is their access to education in effect. And that's what I mean with this. No child will learn to read unless they're taught to read, is the simple fact. So even if heritability was very high, unless somebody teaches you to read, you will not read. Okay. Similarly, in terms of if you think about the number of fingers on your hand, this has low heritability. And this is very bizarre because you kind of imagine surely that is strongly biologically determined as to how many fingers you've got. And it is. But what isn't strongly biologically determined is the variability in the number of fingers on your hand. So there are some people with genetic mutations who grow extra fingers, but most of the variability is caused by people putting their fingers in machines, in effect, and that is the driver of variability. So heritability is about the variance and not the mean. So the mean number of fingers is strongly biologically determined, whereas the variability in the mean is strongly environmentally determined. Okay? And people get this very confused. In effect, it's the same thing about intelligence increasing over the last 3 generations. That is clearly environmental, but intelligence might still be genetic. Because here you're confusing the mean and the variability and these sorts of things, okay? And in effect, when you look at this across various things, you can see that there are some traits such as memory ability that have moderately high heritability. Heritability is on a 0 to 1 scale, basically. Certain acquired disorders like acquired developmental disorders like dyslexia and schizophrenia have much higher heritabilities. And again, these figures get modified all the time as new samples and so on. What about in the brain? Well, you can do this with measures of the brain as well as measures of cognition. And you can show that things like the total brain volume has a very high heritability. This is, for instance, comparing identical and non-identical twins. So identical twins will have very similar brain volumes. Whereas actually other aspects of brain structure are actually much lower in heritability. So the actual total surface area is lower. The grey matter density is highly heritable, but not as strongly heritable as the actual size of the brain. And this is just mapping this in identical twins and non-identical twins. So here what you've got is in identical twins, you're just plotting the degree of correlation in grey matter there. So these pinks show that they have a very high degree of correlation in grey matter between twin 1 and twin 2. And here in twin 1 and twin 2, on average, they tend to have correlations in this kind of purpley range, so 30-40% in the non-identical twins. And the heritability is the difference between the identical and the non-identical. So not the actual correlations, the difference in correlations is what gives you the heritability. And some of these brain regions you can see are basically highly correlated in both of these sets of twins here. And others are less so. So, in effect, heritability says something about the relative degree of environmental versus genetic influence, but it says nothing about what genes are involved or how the genes work and so on. And essentially, the frontier of kind of understanding gene-environment interaction is moving away from this and understanding it at the level of individual genes. The problem with that is that measuring individual genes is not straightforward. There are so many of them that you end up with these massive data sets. There are two broad ways in which people have tried using kind of more molecular genetic techniques to do that, called genotype first and phenotype first approaches. In both cases, it involves getting a sample of genes from people, and this is typically done with these kind of saliva kits that you can put in the post to people. So we do it with people with synesthesia. To get their genome, we just send them these little kits, and they scrape away cheek cells which easily come off and put them in a little test tube. Now here you might think, well, how do you correlate cheek cells with brain and behavior? Well, you can do, because it's exactly the same genome in the cells in your cheek as it is in the cells in your neurons. What differs, of course, is how the genes are turned on that will make them a neuron in the first place. But the actual structure of the genetic code is the same no matter which cell you extract. So at that level, it doesn't matter where you take it from in the body. For certain other things, it is going to matter, and I'll come on to that. But essentially, one thing you can do is that you can then look within the genome at a candidate gene. So you could take, for instance, a dopamine receptor gene and then track differences in the structure of that gene and see how that tracks differences in cognition. Or what's called a phenotype first approach is that you take a behavior, so a cognitive trait, and you search through the whole genome and see which genetic variants are correlated with variations, say memory ability, autistic tendencies, and so on. So these are two different ways of kind of exploring this at the level of molecular genetics rather than at the level of kind of heritability, just looking at differences between twins or adoptive parents. So this is an example of a genotype first approach, and basically what they do is that they take one particular candidate gene. In this case, it's a dopamine receptor gene, D4, that basically exists in two different forms. So in effect, within a group of undergraduates, some people will have a long form, here L, and some people will have a short form. And basically what it means is that that receptor will have a slightly different shape. With this, it will function more or less efficiently. It will process or respond to a dopamine kind of latching onto it in different kind of ways. And the question is, what does that mean for things like personality? Here what you find is that people who have the long dopamine receptor gene score high on a personality trait of novelty seeking or sensation seeking here. So they like, for instance, going on roller coasters. They are more vulnerable to trying drugs. They are kind of interested, oh, I'll have some of that sort of thing, than people with the shorts allele. So they are somewhat more risk-taking and sensation-seeking, I suppose is the way of looking at it. So here people have kind of looked at that from a candidate gene and then looked to see how it affects behavior. The other way of doing this is that you take a candidate behavior and then just search through the whole genome and see what's correlated with it. And this gives you these things here that are called Manhattan plots, because it looks like the New York skyline. On the x-axis here, you've just got the different chromosomes. They're just color-coded. And on the y-axis, you've got your p-value. So here, a p-value, the value of 4 means 10 to the minus 4, or 0.0001. So again, the problem with this is that you've got the problem of multiple comparisons, is that you might get certain things coming out by chance. Yeah.
What are the little squares?
The little squares, each square is a gene.
Oh.
Yes, yeah, no, it's a good question. Yeah, each one is a gene. So what this means is that above this statistical threshold, you've got 1, 2, 3, 4, 5. You've got 6 genes that are correlated at this level of significance with autistic tendencies, in effect. And at this level of significance, you've got that many. And of course, as you go down to levels, everything will kind of, come out. Yeah, but that's a good question. Each little square is a different gene, in effect, within the chromosome. So basically, these are the alternative kind of approaches of doing this. And at the moment, there are kind of international efforts to kind of document this and link things to behaviour. So we've got the UK Biobank, which literally has genetic material from 500,000 people, and they follow their medical history. 100,000 people are having MRI scans of their brain, and you can ultimately link together this. And you can go to the website and download 100,000 brains, or you will be able to. How your computer will process that, I don't know, but all their genomes are online. All their mental health information is kind of online, obviously anonymised. And this is kind of the future of how people are going to look at gene-environment interactions and figure out how it's going to go to the brain. But the statistics behind this are pretty mind-boggling, as you would imagine. But what kind of things would you look for in these samples, or how would you kind of understand how, for instance, these kind of genes are kind of operating and affecting behaviour? So genes and environments can interact in various ways, and these are the three kind of big ones that I'll talk about. One is epigenetics, and this is the idea that your environment alters your gene expression, so not your genetic code, but the way that the genes are switched on and off. GeneEnvironment Correlation, this means that people with a certain genetic predisposition seek out certain environments. And GeneEnvironment Interaction, which means that basically your cognition and your behavior isn't to do with your genes, and it isn't to do with your environments, it's to do with your gene and your environment in combination. And having one of those things isn't enough, you need both. It's like a double risk, in effect. You've got a risk in your environment and a risk in your genes, but you will only then become vulnerable if you have, in effect, a double hit, would be one way of putting it. So what is epigenetics? So it's basically changes in the way that the DNA code is read off, but not changes in the DNA code itself. So your DNA code is this double helix thing, which is wrapped around proteins when it's stored in the nucleus. And in effect, what happens to the genetic code is that you can attach little markers on it. So these are just little chemical compounds. So methyl is one. There are other groups that do it. And in effect, what adding these molecules does is that it kind of makes the DNA more or less accessible to be read off. It actually affects how tightly bound it is to these large balls here. So it will either loosen it or tighten it on, so it makes it easier or hard to read the genetic code. And basically, adding these things here can depend on your environment. Basically. So for instance, a good example of this is a stress response that happens again in a sensitive period early on. I should also say that this here, the epigenetic marking, you have to measure in neurons. So it's not clear that you would be able to detect what's going on in your neurons by getting at it from the cheek cells. So here, obviously, the main focus is animal models or post-mortem brains in humans, because that's the only way of knowing about this. Obviously, you can't extract them from biopsies. If you look at rats, basically rats differ in their level of caregiving. So caregiving in a rat depends on how much they would kind of lick and groom and cuddle up to their offspring. And in effect, you will get a kind of a normal distribution of caregiving. What you find is that pups who are born to neglectful mothers who don't give them much care have a heightened stress response that survives into adulthood. And this stress response is linked to an epigenetic change in a receptor gene that's part of the stress pathway in the brain. So glucocorticoid receptor responds to cortisol and these sorts of stress hormones. So it's not that their genes are altered or that these mothers are genetically different. It is to do with, in effect, the babies developing a stress response, and their coping mechanism is to change their expression of the genetic code. It alters that, and it's a lifelong change that they have. And people have kind of looked at this, and it does depend on certain time windows. In humans, it's also been looked at, in this case, in suicide victims. So these are adults who've committed suicide, and versus controls who died of other causes. And basically, you can look at these epigenetic markings in the brain post-mortem, in particular regions of the brain as well. And this is some kind of measure of the glucocorticoid receptor. I was trying to figure out why it was lower in the critical group, but basically they are switching off that certain gene. It's as if their coping mechanism is to down-regulate a natural stress response. What you find is that basically these epigenetic changes are not to do with committing suicide. They are to do with whether or not you were abused 20 years earlier in life. And this is various aspects of abuse. So neglectful mothers who were drug addicts or whatever, physical abuse, sexual abuse, and so on. So obviously, people who commit suicide typically have mental health problems. So it isn't to do with mental health problems in themselves. It is to do with things that happened early in childhood. People have looked at this because whether or not these kind of epigenetic markers can carry across generations. The feeling is that they kind of get washed out, but maybe not in a single generation. So in effect, if you had a bad childhood, there's some evidence that they can be passed on in the egg or the sperm, in effect, in those sense. But they are not permanent changes. They are seen as being reversible. People used to think that they were reversed as soon as you gave birth and so on, but they seem to be there in a weaker form. So that's kind of epigenetic marking. Gene-environment correlations, well, we've already seen one example of this. And this is the dopamine receptor gene, or the long variant that's linked to risk-taking. So what a gene-environment correlation means is that if you have a certain genotype, then you are more likely to seek out certain environments. So people always imagine that what happens in your environment and what happens in your gene are just two separate levels. But the idea here is that they're not separate levels. You create your own environment, either through being intelligent or being stressed or being a risk taker or so on. That in effect, a lot of things that people used to think were environment are probably your genes just coming out in a different way. And there's whole kind of ideas behind this, that what used to be labelled as environment is genes in disguise, in effect. Here there are people who study this. So there's the Rudd Center looks at kind of adopted children, and they do a lot of these kind of modelings here to see whether or not it is to do with the child's genotype or the parent's genotype or something in the environment that's done it. But here this is looking at parenting style. So traditionally you would imagine that parenting style is something to do with the parents, and of course the parents are involved in that. But the idea here is that the child's genotype might encourage a particular style of parenting. For instance, if they have challenging behaviour, tantrums or whatever, they don't sleep, that the parents will put into place, for instance, more relaxed or more kind of authoritarian parenting styles. So here, this is in effect that the child's genotype is creating, in effect, ripples in how other people kind of interact with it that will almost reinforce its own genotype or try and constrain it. So here, this is what's shown here. And of course, I'm not saying that the parents' behaviour and the parent's genotype don't matter, but you can statistically separate them out and show that the child's genotype also matters as well. The child will create its own environment to some extent. What about gene-environment interactions? So a gene-environment interaction is basically that you develop a clinical vulnerability because you have a certain gene and because you have a certain environment. And neither the environment's causal nor the gene's causal. You need the combination of the two. So the classic studies that looked at this were carried out in the early 2000s by this particular group here, partly based in the Institute of Psychiatry in London, but looking at a sample of births from Dunedin in New Zealand, which I think was it in the 1980s? These people are in the 40s now. Yes, it must be about then. And basically what they did is they took 1,000 consecutive births in this time, and they looked, for instance, at their attachment styles at infants. They looked at their cognitive development at school. They looked at their worries when they were children. They looked at whether they smoked dope in their teenage years. They did all of these things longitudinally. And they've had a very low dropout rate. So this cohort is still being studied. I don't know how many are left, but it's certainly over 900. So they still have most of the sample actively taking part in this research. And at the time, 1,000 was seen as being a very large figure. Now it's seen as being a very small figure in this field, and this is one of the problems that's emerged in interpreting this literature. But in effect, they reported a number of effects, which I won't go through in detail, but I will give you a flavour of them. So this study here looked at what's called a monoamine oxidase A. This is a gene that's involved in metabolizing I think various neurotransmitters that exist in multiple forms. You can also Google it as the warrior gene. That's a label that's been given to it. And basically what's found is that if you have a poor childhood, that you're kind of neglected or maltreated in some way, plus you have a particular variant in this gene, you are far more likely to have antisocial behavior. So what this means is that the whole is greater than the sum of the parts, in effect. So neither having this gene in isolation nor having a poor environment as a child is sufficient. It's having the two that kind of pushes you up to another level, in effect. What's also been shown is that this is, so 5-HT is serotonin. This is a serotonin transporter gene that exists in short and long forms. So some people have short and some people have long. And basically what you find is that if you have one of those genes and bad things happen to you in life, not necessarily in childhood in this case, and so that you are more likely to become depressed and develop suicidal thoughts, in this case, for instance. And this gene here, so COMT, is involved in dopamine. And the claim here is that if you were to smoke cannabis in a sensitive period in adolescence, not so much in later life, that you are more likely to develop schizophrenic symptoms. So again here, it's not just the simple tabloid claim that smoking cannabis gives you schizophrenia, nor that schizophrenia is genetically mediated. It's just that some people have a risk, and that then kind of amplifies that risk if you have it. So, and again, this kind of conforms with people saying that many people smoke cannabis and never go on to develop schizophrenia. So it's probabilistic, but it's probably a weighted probability. And this is in effect the data that was reported here. So the COMT gene exists in two forms. One is called VAL and one is called MET. These are just amino acids that are encoded by the gene. And basically, if you have the VAL gene, you have two copies of it, because you get a copy from your mother, a copy from your father, and you smoked cannabis in adolescence, you are far more likely to go on to report schizophrenic-like symptoms. They can look at various things with this. So because it was longitudinal, so they can go back and look at these kids when they were 11 and see whether or not there were kind of other early markers of this and so on. So again, it's a very nice database. But actually these results have turned out to be rather controversial, not least because of any kind of sociological kind of implications, but just in terms of the science. But it's very easy to see why these results had a big impact, because in the days of heritability, all you would say about schizophrenia is that it's half nature, half nurture. That's the conclusion of your essay. Here you can actually move away from the half nature, half nurture and actually have a more sophisticated kind of mechanistic argument as to how nature and nurture are actually interacting and showing that they are both there. But the reason why they've been controversial is that here these were taking what's called the candidate gene approach, is that researchers looked for one particular gene and then went out and found that there are these differences. This has been overtaken by what's called GWAS. So this is where you search through the whole genome. So you search through every single gene and see which genes are related to antisocial behavior or depression or schizophrenia. And what you hope is that these will converge. These initial genes that people decided to look at should pop out when you look at this. And these approaches, again, typically have tens of thousands of people, and because people are pooling genes across the world, they're now in the hundreds of thousands. And some of these early genes are not appearing in this. And this has kind of led to a bit of kind of worry, for obvious reasons. It's not the case that these never replicate. There are interesting replications of this. And people have looked, for instance, at the brain structure of people who have these different variations and shown them things like angry faces and happy faces, and shown that people who are vulnerable to antisocial behaviours respond very strongly to angry faces. So you can do these experimental techniques, but they do not consistently come out in these large-scale approaches. So there are two kind of answers to this. One is to throw out all the old results and say, hey ho, this is just, the way things sometimes go in science, that we get things wrong. And that is possible. There are some people kind of advocating that. Other people have taken another approach and say, well, actually, it still might depend on how you're measuring the environment. So even if your sample goes from 1,000 to 100,000, if you are not measuring the environment properly and all you're doing is just throwing in these genes, for instance, and seeing what pops out, then if you're not asking the right questions, it doesn't matter what your sample size is. And that's been the critique or the pushback from the people who did this original research, is that they said they documented the environment very carefully and plotted when exactly in life these kind of triggers were present. And the other studies are taking a much bigger, broader brush approach in the hope that simply having more data is going to reveal the answer. And it might not if you don't ask the right questions. The other kind of thing that people have said as to kind of rescue the original data sets is that they're not asking the right environment questions. There might be sensitive periods in which the environment kicks in, but also that there might be it's not always the case that these genes convey a negative vulnerability. They might also convey a positive vulnerability that again is not captured in there. What does a positive vulnerability mean? Basically, it just means that some people are more susceptible to things happening in their environment, whether it is good or bad. And other people, you can throw anything at them and their environment, they will carry on the same. They are just, not very plastic in terms of their behaviour, I suppose. And this is the kind of evidence that's been looked at this. So this is from Birkbeck in London, a group there that's looked at parenting quality here. And here, I think these are adolescents looking at their emotional self-control, their ability to regulate their emotions. What you've got on the x-axis here is parenting quality. So the people in the middle have average parenting quality. The people down here have unusually bad quality of parenting. But on the other side, these people have unusually good quality of parenting. I'll be interested to know what that is, but anyway, they have measures of these things. And what they then look at is that they look across multiple genes here. They looked at 5 genes that are involved in kind of sensitivity to particular behaviours, including actually 2 of the original 3 that were looked at in the studies I reported there. So again, this is a partial replication. And what they find here is that basically if you've got a lot of these kinds of genes and you have bad parenting, sure enough, it leads to a negative outcome for you in terms of your ability to regulate your emotions. But what's also found is that those people have unusually good parenting and exactly these same vulnerability genes have better than average this. So it suggests that these people aren't just vulnerable to negative things, they're just vulnerable to any variation, whether it's unusually good or unusually bad. And again, a lot of these very large GWAS studies are only looking at this end of the distribution, those people who are neglected, those people who are unusually depressed, and so on. Whereas people who have bad life events thrown at them and are unusually happy are an equally important kind of group. And that, I suppose these people here, for instance, would be people who had very poor parents, but hey, this had no effect on them. For instance. So these are the kind of ways in which we can kind of resolve these discrepancies. But ultimately, the aim is that both of these genetic approaches should converge when we understand them at a more mechanistic level. Any questions? Yeah.
How do you measure parent equality?
Yes, I was wondering that, as I was saying. I think, so the question here, I suppose, is whether you're asking the parents or asking the children, and that's a very big difference, isn't it? But that is, it will be a questionnaire measure, almost certainly, in here. But who you're asking, I would have to check. Yeah. If they're asking the actual recipients, then that's a little bit of a confound. Although but the people are blind to their own genetic makeup, so they can't use that to influence their decision. So to summarise, the genes, or kind of nature, provides a guide for how the brain will develop, but it does not provide a detailed blueprint. It's just almost like a recipe book that you can then do what you want with it. So it anticipates the presence of certain inputs in the environment. So, for instance, it expects to receive vision, it expects to receive language, and at particular stages of development. So this is the classic kind of notion of sensitive periods. But genes and environment work together. It's not that the genes kind of create the brain and then the environment does everything after. And we can evidence this by epigenetics. So literally the genes get switched on and off by the environment. The genes can create certain environments, so people with certain genotypes will seek out certain environments for themselves. And the genes and environments can interact to create these kind of vulnerabilities with this. And I think that the thing that we are kind of lacking at here is basically how genes work together. So this kind of study here, it's unlikely that there'll be single genes that are coding for depression. Nobody really believes that. And in a way, the old-fashioned heritability estimates were just summing together all the genetic effects. And now the question is, now people are looking at individual genes, how will they explain something when there's likely to be so many different pathways that are doing that?